Okay, in this tutorial I'm going to show you, use another little animation to try and explain to you what enzyme inhibitors are and the different types of inhibitors. So enzyme inhibitors, there are basically two types of inhibitors. One is called a competitive and the other is a non-competitive inhibitor. So a competitive inhibitor mimics the substrate and binds the active site. So here a competitive inhibitor binding to the active site competing with the substrate. A non-competitive inhibitor would bind to a regulatory region which changes the shape of the enzyme's active site so that the substrate can no longer bind. Now one thing that I don't like about this animation, or one thing that's maybe not quite shown so well, is normally the inhibitor has a similar structure or shape to the substrate. So normally a competitive inhibitor is actually a molecule that is very similar in shape and structure to the substrate. And that's one of the reasons why it can actually bind to the active site. So imagine sort of like if I took my key and stuck it into a lock, and if it was a very similar key, it might not actually turn the lock, but if I kind of broke it off in the lock, you wouldn't actually be able to get your key in there. So that's kind of what a competitive inhibitor does, is it binds to the active site and usually has a very similar shape to the regular substrate. But for some reason, usually what happens is the competitive inhibitor isn't broken down or doesn't undergo the same reaction, or takes a lot longer to be broken down or undergo the reaction than the regular substrate. So that's why it kind of blocks up and prevents that enzyme from actually carrying out its job. So a non-competitive inhibitor binds somewhere else to the enzyme and changes its shape. So we looked at proteins earlier in this course, and proteins are made up of those amino acids, and it's the shape of the protein that the amino acids fold up into, so that the protein's precise 3D shape is what really determines its function. So this enzyme, the active site, the shape of this enzyme determines its function, and the inhibitor here will actually change the enzyme's function. Now you may have looked at the idea of a denaturing an enzyme. So if we heat an enzyme or a protein, we'll actually disrupt bonding between the amino acids and it will unfold, it will denature, and it won't work properly anymore. This is a little bit different because the binding of the inhibitor here is often reversible. So there may be something that will cause the inhibitor to be released, or maybe the inhibitor will break down after a certain amount of time. And when that happens, usually, in most cases, when the inhibitor is released, the shape of the active site will be restored. So an inhibitor here, it usually results in a shape change and a change in the conformation of the enzyme, but it's normally not a permanent shape change, and that's what makes it different from denaturing. Now we'll look a little bit more at denaturing in one of the other tutorials. But just quickly again, competitive inhibitor binds to the active site, a competitive inhibitor binding to the active site, and a non-competitive inhibitor binding to a regulatory region. So with the competitive inhibitor here, you can see competing with the substrate, non-competitive here binding to a regulatory region, and essentially changing the shape of the active site. Now with a competitive inhibitor, because the inhibitor is actually competing with the substrate, what that often means is that if we add a whole lot more substrate, we can overcome the effects of the inhibitor. Because if we really flood the system with a whole lot of substrate, the probability of a substrate molecule binding instead of the inhibitor is much more likely, and so we get the vast majority of the reaction occurring as per normal. So we can actually outcompete the inhibitor by flooding the system with a whole lot more substrate. Now apparently some snake venoms or snake bites essentially act as an inhibitor for certain enzymes, and one thing you can do if it's a competitive inhibitor, you can flood the system with normal substrate and it will overcome so whatever the enzyme normally acts on, just pump a whole lot of that into the system, and then essentially you'll outcompete the inhibitor and restore the reaction, the function that was happening beforehand. However, if the venom is a non-competitive inhibitor, if there's something in the venom that binds to an enzyme and changes its shape, then it doesn't matter how much substrate you throw in there. It's not going to make any difference because the active site is actually free, but it just can't bind because it's now a different shape. So that's the idea of inhibitors. So inhibitors, competitive and non-competitive. One thing to be aware of is that in the Cambridge course, technically, if an inhibitor binds to the active site permanently, then it's technically actually considered a non-competitive inhibitor. Now the reason for that is that if the inhibitor binds permanently to the active site, it's actually not going to matter how much substrate we throw at it. It's not going to be able to outcompete the inhibitor. There may be some initial competition, but once the inhibitor is bound, it'll be bound permanently, and it's only a matter of time then before all of the enzymes are blocked by an inhibitor. So if I just go quickly one more time here. Two types of inhibitors. Alright, just wait for it to go through. A competitive inhibitor, which mimics the substrate, and a non-competitive inhibitor. A non-competitive inhibitor, which binds to a regulatory region. Alright, there we go. Alright, so fantastic. Competitive is usually a case where we get an inhibitor binding to the active site. But if this inhibitor binds permanently, then it's actually considered a form of non-competitive inhibition. The reason being, in most cases what happens is the inhibitor here will bind for a certain period of time to the enzyme, and eventually it will come out. And so at that point, there's a probability as to whether the normal substrate will bind to the enzyme or whether the inhibitor will simply bind the enzyme again and block it again. And that probability obviously is based on, partly at least, how many molecules, what the relative concentration of the inhibitor and the normal substrate is. So by flooding the system with a whole lot of extra substrate, whenever one of the inhibitors pops out, chances are one of the substrate molecules will jump in and fill the space, and the enzyme will carry on doing what it normally does. So by flooding the system with substrate, we can outcompete that inhibitor. But if this inhibitor binds permanently, then it's only a matter of time before eventually all the enzymes are blocked by an inhibitor, and it doesn't matter how much substrate we throw at it, it's blocked permanently now because it's not going to come out. Now normally I think there's probably no such thing as an inhibitor that actually probably binds forever. Chemicals eventually will degrade or break down or be metabolized, things like that, but it'll essentially probably bind so long that it'll be bound for the lifetime of the enzyme. So the enzymes have a limited lifespan. Eventually they get broken down, or they get degraded, or they just wear out. They break down on their own, but often they get recycled by the cell. So competitive inhibition is an inhibitor that binds temporarily to the active site, and by throwing a lot of substrate at it, we might be able to outcompete that. If it binds permanently, it's actually considered non-competitive inhibition. Now any inhibitor, whether it binds permanently or not, is non-competitive because it doesn't matter how much substrate we throw at it. The active site is actually technically free, but it's just changed shape, and so the substrate doesn't fit anymore. So whether it binds permanently or not, we also sometimes use the terms that means whether it's reversible or irreversible. So competitive inhibitors binding temporarily to the active site, and non-competitive either binding to a regulatory region or binding to the active site permanently is technically a non-competitive form too. So guys, I hope you found that helpful, and I'll try and get on with the next one as soon as possible.